In the arid deserts of the American Southwest and parts of Mexico, a quiet threat is stirred up with every gust of wind. Our presentation is on valley fever, which is a fungal infection caused by Coccidioides mitis. When spores from this fungus are inhaled, they can infect the lungs, leading to flu-like symptoms, chronic pulmonary complications, or, in severe cases, life-threatening systemic disease. Despite nearly a century of research, no vaccine exists for this disease. Treatment options are limited and often ineffective in preventing long term complications. Mitis has a dual life. In the environment, it grows as mycelia, but once inside a host, it transforms into spherules, large parasitic structures that produce hundreds of endospores. It's during this stage that the fungus is most dangerous and where the host immune system must respond to fight infection. That makes it a prime target for vaccine discovery. Our project aims to identify a highly expressed, surface exposed antigen that's upregulated during the parasitic spheral stage, a candidate for developing a subunit vaccine for valley fever. To do this, we applied a functional genomics pipeline using transcriptomic and structural tools to sift through potential targets and narrow in on the most promising ones. Began with RNA sequence data from the NCBI geodataset GSE 171286, which compares gene expression in mycelia, young spherules, and mature spherules of Mitis. Using DE sequence 2, we performed differential expression analysis to identify genes that were significantly upregulated in spherules compared to mycelia. These are the genes that are most likely to be involved during the infection. Next, we predicted the 3D structure of these candidate proteins using AlphaFold, a deep learning model that reveals how proteins fold in space. We focused on proteins with exposed loop regions, which are accessible to the immune system and likely to interact with antibodies. We then use IEDB's BepiPred 2.0 to predict linear B cell epitopes, short regions that the immune system can recognize and mount a response against. To better understand these candidates, we annotated them using KEGG and BLAST to assess their biological roles and conservation across other species. Surprisingly, some of the most promising genes had limited functional annotations, suggesting novelty or species specific expression. To bring it all together, we used ggplot2 in R to generate volcano plots, heat maps, and expression profiles that visually highlight key findings. One gene clearly stood out, CIMG 04613, also known as SAUGP, spheral outer wall glycoprotein. This gene showed a log2 fold change greater than 8 and an extremely low adjusted p-value, indicating it is strongly upregulated in the parasitic stage. Expression analysis confirmed minimal activity in the mycelial stage and intense activation in both young and mature spherules, especially during the early parasitic stage where immune intervention would be most effective. The AlphaFold model of SALGP revealed a folded loop-rich structure, ideal for antigen antibody interactions. Additionally, epitope mapping using BepiPred predicted two strong linear B cell epitopes, including a 104 residue region spanning an exposed section of the protein surface. Interestingly, SALGP had limited annotation in KEGG or Reactome databases, suggesting it might be species specific, which could reduce the risk of off target effects in humans. This study demonstrates how functional genomics can rapidly identify stage specific antigens for vaccine development. From differential expression to epitope mapping, our analysis highlights SALGP as a compelling vaccine candidate, highly expressed, surface accessible, and immunologically relevant during the infectious stage of CMItis. Future work will focus on vivo validation and immune response testing, but this pipeline lays a strong foundation for developing a subunit or recombinant vaccine against valley fever powered by genomics.